I'd like to start by thanking Ross and the New Zealand Drug Foundation for inviting me over from Australia and congratulations on what has been a fantastic conference. So I thought I had half an hour, but I've been informed I've got 20 minutes, so excuse me, I'm going to have to flick through some slides rather quickly. I was going to start with the regulatory context, but Wayne's already pretty much covered that. So the take-home message from this slide is the last bit at the very bottom, which says, if we want to approve Sativex for any other condition than multiple sclerosis, we'll have to demonstrate that it's safe and it has efficacy for other conditions. So, to the point of my talk, why would we be looking at Sativex for cannabis withdrawal? Well, we're looking at Australian data here from our National Drug Strategy Household Survey, and the colours of the bars are the different years that we do the survey, and you can see the different drug types across the bottom there, and the y-axis is the percentage of the population who consume that drug type, and as you can see, cannabis consistently for many, many years, the most prevalent illicit drug in Australia. We also have quite a nice data system in Australia on our drug and alcohol treatment episodes. It's called the National Minimum Data Set. Basically, anybody who goes into one of our drug and alcohol treatment centres in Australia gets an episode recorded, and we do that every year, and we collect all sort of data, such as what was the primary drug of concern that they presented to the treatment centre for. And that's what we're looking at here for just 1 year, 2008, 2009. And there you can see cannabis, the second most prevalent reason for presentations to alcohol and other drug treatment services. So irrespective of your position on whether it should be legalised, whether it's harmful or not, this is the hard data. People are asking for help with their cannabis use. About 33,000 people in that 1 year. And that trend for people increasingly seeking help for the cannabis use has been increasing over time. Here, this is actually European data, but the point I'm trying to illustrate is that treatment seeking is increasing, and it is doing so all over the world. In Australia, it went from about 2% of treatment episodes back in 1990 were for cannabis as the primary drug of concern to about 22% in the last recorded episode. So it's very prevalent and people are asking for help with it. So what do we do when somebody walks through our door and asks for help with their cannabis use? Well, as with all drug problems, there's two major areas we tend to focus on, either withdrawal management to help them with the acute phase of getting off the drug or promoting long-term abstinence. In this talk, I'm dealing with only withdrawal management for cannabis. So what do we know about cannabis withdrawal? Well, until recently, not a lot. Although there has been a lot of research taking place over the last decade or so. Here's just 4 papers. There are now many more of people exploring the withdrawal syndrome when people stop smoking cannabis in treatment seekers, in non-treatment seekers. And down in your bottom right-hand corner, there's one comparing cannabis withdrawal to tobacco withdrawal and looking at how severe it is and what sort of contribution it makes to relapse. And that last point, the contribution that cannabis withdrawal makes to relapse, is the most important point, because within that point lies the clinical significance of it. If somebody's trying to get off cannabis and the withdrawal symptoms are driving them back to use it, well, that's a reason that we need to look at it and try to do something about it. Sum total of this research has resulted in the cannabis withdrawal syndrome being included for the first time ever in the DSM, the new DSM, released in 2013. So it's all quite timely. We've now got a new disorder, that we need to find a treatment for, and as you probably know, there's no approved pharmacotherapies for treating cannabis dependence or withdrawal. People tend to give symptomatic medication relief in the form of benzodiazepines or antidepressants or whatever, but there's no evidence for any of them. That's not to say there hasn't been research in the area. So this rather busy slide just gives you an overview of the research into pharmacotherapies for cannabis withdrawal over the years. You've got the drug over in your left-hand column, what type of drug it is, what its action is, and the paper, the reference to the paper for the study. So I'm not going to dwell on it, just to say that there's been quite a lot of work. It's mostly been small-scale, open-label studies in laboratory setting with non-treatment seeking people, and not a whole lot of efficacy has been proved with pretty much any of them. Rimonabant is worth mentioning at the top, the antagonist, a potential antidote to being stoned, I suppose. Blocks the CB1 receptor, and that did show quite a lot of promise as a medication, but it was removed from the market because of adverse psychiatric consequences. But what I'm going to focus on is the bottom three here, which is the CB1 receptor agonist. And this is the point of the talk today, is cannabinoid replacement therapy. And we've heard a lot about these drugs from Wayne and previous speakers already. And I'm just going to give you a bit of background to the work that's been done on the agonist replacement therapy. So this is where it all begins. You've got the THC in the smoked cannabis, and we're trying to find an effective pharmacological substitution for that drug at the CB1 receptor agonist. So most of the work in this area has been done with dronabinol. It's a synthetic THC drug made in the laboratory, packaged into a tablet. You swallow it, it gets digested by your stomach, goes through the bloodstream to the brain. Obviously a radically different pharmacokinetic profile to smoked cannabis, and as Wayne's already covered for me, people didn't really like it as much as cannabis. But in the trials that have been done, it was found to reduce withdrawal symptoms, but it was only done in non-treatment seekers, and that was in a laboratory context. When it was taken out into the field with treatment-seeking cannabis users in an outpatient environment with large numbers, about 150 people or 200 people, who were using it for, I think it was about a 6-month period, it did not reduce cannabis use. More recently, another synthetic THC analogue, I suppose, nabilone, has been explored. This was only published this year, so it's all quite new. It was found to reduce withdrawal symptoms, and it did reduce cannabis relapse, a measure of cannabis relapse in non-treatment seekers, but it was in a very tightly controlled artificial laboratory environment in America, in New York. They've got this laboratory where they lock people up for a month and they give them access to placebo and active cannabis joints, and they're either on placebo or active nabilone, and they basically give them some money at the start of the study, and they can choose whether or not to spend their money on accessing a cannabis joint, and that's their measure of relapse. Do they choose to spend their money? And it did reduce that measure of cannabis relapse. So that's promising, but it's yet to be taken out into a clinical environment. And so what we're looking at here, have tried to depict, is a sort of increasing gradient of bioavailability in these drugs. Nabilone is more bioavailable than dronabinol. It means it gets into the blood system quicker, it gets to the brain quicker, and it appears to be slightly more efficacious. And that's where Sativex sits in the middle. It's got the new route of administration. It's sprayed into the mouth. It's absorbed through the oral mucosa. It gets in faster and it gets to the brain quicker. And we thought these properties mean it would have would be a better drug for this indication. You'll notice also that there's two molecules rather than just the THC molecule. That's the cannabidiol, which we've heard a lot about, so I'm not going to dwell on it, just to say that for this particular indication, the fact that it has been demonstrated to reduce THC-induced paranoia makes it particularly interesting, because what it means is it reduces the abuse potential. If that is the case, it means we can give it to this population without worrying too much about it being abused. Made by GW Pharmaceuticals, and just to lighten the mood a little bit, I'll give you a little snapshot inside GW Pharmaceuticals' grow setup. I was lucky enough to visit, and just they've got all these different rooms with the plants at different stages of development, the clone chamber, and here I am in the middle of the grow setup. And here's one of their cannabidiol plants. So just so you know, they don't breed plants that are high in both THC and high in cannabidiol. They breed separate plants. This one is one of their cannabidiol ones, and they extract the active compound and then mix it together. Just on the cannabidiol point and why is it potentially important, here's some data, some real data from cannabis seizures from the police in New South Wales, Australia, from some colleagues of mine. 250 police seizures of cannabis were taken into the psychopharmacology laboratory at Sydney Uni, and they just measured them in their machine. And you can see the top graph shows the about 15% THC on average across all those 250 seizures, and the bottom graph is the cannabidiol. And you can see over on the right is the one you want to look at, the CBD total, practically non-existent in real cannabis on the streets. So what did we do in our study? Well, we went out to treatment centres in Sydney and Newcastle in Australia, and we put up these posters to recruit people who were over 18 years of age, regular cannabis users with a desire to quit. They had to have tried to quit and failed before, and had to be prepared to go into a hospital inpatient unit for 8 nights, which seems rather excessive for a cannabis withdrawal treatment, but that's the nature of the beast with an experimental new product delivery, you know, so we would never have got it funded if we didn't do it that way. I had a video which is a news clip from Newcastle that we were using for recruitment, but because of the time constraints, I'm not going to show it. So we recruited them, we randomized them to a double-blind placebo-controlled trial, and this is who they are. So we got 51 people in total, and they were an average of 35 years of age. 76% of them were male, and they smoked an average of 23 grams of cannabis a week, and that is an average. So by definition, some of the people were smoking a lot more than that, some a lot less. It seems like quite a lot to me. And they've been smoking for 20 years on average. And they scored an average of 12 on the Cannabis Severity of Dependence scale, which is a 15-point scale. Scores of 8 or above are in the clinical treatment-seeking range. So a heavy using, long-term using, severely dependent people looking for help with their cannabis use. This is the dosing regime that we used, and this is the nice thing you can do with a pharmaceutical product like this. You can really clearly identify what dose you're delivering and what effect it has. So across the bottom there, you can see the number of sprays. So it was 8 sprays we delivered 2 times a day, and then we ramped up to the highest dose of 8 sprays 4 times a day, and then a rapid drop-off to day 7. And then to day 6, and then a 3-day washout period. And that equates to a peak dose of about 80 milligrams of THC, 80 milligrams of CBD at peak dose. And you can see the sample sizes in each group there, 27 in the Sativex group, 24 in the placebo group. We took blood samples from them at 3 time points before they had any medication, at peak dosing and after, to check the cannabinoids. This is the cannabidiol in the blood. So you can see, as was shown in the illicit cannabis seizures on day 1 before they had any medication, nobody's got any cannabidiol in the system. The Sativex group, we increased the cannabidiol and it dropped down with the decrease in dose. Oops, sorry. And this is THC. Nice pharmacological substitution there. So we managed to maintain the blood levels of THC to peak dosing from what they came into the study with, and then it dropped down to placebo levels at day 7. So to move on to the behavioural outcomes, this is the cannabis withdrawal scale that we've been developing at UNSW over the last few years. It's a 19-item measure. You can have a look at the symptoms yourself. People rate them every 24 hours, reflecting on the last 24 hours, what severity on a scale of 0 to 10 they've experienced for each of these items. And then you can take the average across all 19 items, and you get a number from 0 to 10. Which is the severity of total cannabis withdrawal scores. You can also group items within this to get the DSM-5 withdrawal symptoms, which I'll show you shortly. So this is the punchline, I suppose. This is the withdrawal scores, overall cannabis withdrawal scores up the left-hand axis, days in treatment along the x-axis, and the vertical line there shows the day when medication ended. The asterisks show statistical significance between the difference between the days. And we're measuring withdrawal each day relative to the baseline, so what they experienced when they were smoking cannabis on the day they came into the study. So nice separation during medication delivery, but not significant after we stopped medication. I will point out, whilst I'm on the slide, because somebody always comes back to me on this, it is a very mild withdrawal syndrome. So it's a 10-point scale, but we're only seeing average withdrawal scores in the placebo group of about 1. That is on average, and when you unpack it, there are some people experiencing quite severe withdrawal. But the point I want to make is it is mild, but it still has been shown to contribute to relapse. So like nicotine withdrawal, reasonably mild, but still worthy of looking into it. And underneath that overall cannabis withdrawal score, you've got the DSM-5, the separate DSM-5 withdrawal symptoms. I'm not going to work through them all. I'll just let you have a look at the different ones: craving, irritability, decreased appetite. The point, I suppose, is that they do have a slightly different profile, and not all of them were significantly suppressed, but for all of them, the green line, the people receiving the Sativex, is below the placebo group. So we've got pretty strong evidence of efficacy for dampening withdrawal symptoms there. Another one of our major outcome measures was retention in treatment. Up the left-hand axis, you've got the percentage of patients still in treatment at each day of the 9-day inpatient stay. You can see the black line, the placebo line. By the day 6, the last day of medication, 50% of the people on placebo had said, "I've had enough of this, I'm going home." We still had 85% of the people on Sativex in the study. That did drop off dramatically straight away after we stopped giving them the medication. Days 7 to 9, no difference between the groups. They all went home. But still an important finding. We've heard quite a lot over the last 3 days about the difficulty in retaining these patients in treatment. So that's potentially quite important. And the intoxication scores, we measured them each 4 times a day. We dosed them, and we measured them 4 times a day for intoxication. We measured them before and after we dosed them, and as you can see, not significantly different, even though slightly elevated in the Sativex group, not significantly different. We also did what's called testing the blind. We asked them, it sounds more complicated than it is, we just asked them before they left the study, before they left the centre, what did they think they were on? And that's across the bottom there, and the colour of the bars is what they were actually on. So as you can see, 60% of the people said they were not sure, and basically the take-home message is they did not know what they were on, even though they were receiving the highest dose that has ever been given to humans. They couldn't tell what they were on. Although I do wish I hadn't given them the option of saying not sure, and force them into a choice. But hindsight's a wonderful thing. Adverse events. So we got this long list of adverse events from the GW Pharmaceuticals protocol, and they were not significantly different between placebo and Sativex. The bad news, long-term outcomes were not improved by the drug. So this is I say long-term, it's not really long-term. 28 days after they left the unit, we brought them in and interviewed them, measured their urine, see what the cannabinoid levels were like. And you're just looking at the green bars are those people on Sativex, the dark bars are placebo. Both groups dramatically decreased all measures of cannabis-related measures, but not significantly different between the groups. So the take-home message is Sativex significantly decreased withdrawal-related anger, irritability, depression, loss of appetite, cravings to smoke cannabis. Importantly, it decreased the overall cannabis withdrawal syndrome. It retained people in treatment significantly longer, and they were not more intoxicated. However, a short course of Sativex over the acute withdrawal period does not lead to better outcomes after withdrawal management. Where to from here? Well, we're trying to get a longer-term maintenance trial up and running to test for relapse prevention, but the grant wasn't funded this time around. And we're thinking about possible extensions into treatment for comorbid pain and cannabis use and comorbid PTSD and cannabis use. I would like to acknowledge the investigative team and the NHMRC for funding and GW for providing the medicines and the placebo and the nursing staff at each of the units. Thank you.
Questions? Down the front here.
Thanks, David. Question on the last finding that you had about not showing much effect after 28 days. How does that compare with the data in Australia with methadone, when that stopped, when folks stopped getting methadone, and then after 28 days, what they're seeing?
Well, I'm not qualified to talk about that, I'll be honest. Maybe Wayne might know more about it, but thinking on my feet, I would say that it's probably highly dependent upon the context that you're talking about. There's a lot of different contexts for methadone treatment. People have been on it for 20 years. You know, the experiments where you're looking at the outcomes for methadone treatment were done a long time ago. And I suspect it's the same deal. You know, I mean, it's acute withdrawal management is not a treatment for the drug problem.
Sir, over there.
That was slightly backed up by a recent buprenorphine study that looked at short-term versus slightly longer extended withdrawal and the outcomes. And the extended did have some benefit. So your idea that you might have a slightly extended, a trial of extended,
yes,
might bear some benefit.
Of course, but then you're talking about moving into a cannabis maintenance therapy, you know, like methadone maintenance, and you know, I mean, this is the debate, I suppose, that we're going to see soon. I mean, is that really a route that we want to go down? And well, we'll see.
Yeah, David, thanks for the presentation, very interesting. Going back to an earlier slide where you were looking at the numbers of people in treatment presenting specifically for cannabis.
Yes.
Is around about 33,000 for that 2009, I think it was.
Yeah.
Any idea of what proportion of those would have been pushed into treatment, you know, coerced into treatment through the courts versus people who are actually presenting, you know, voluntarily?
No idea, I'm afraid, although I think we've already heard, possibly from Steve Allsop over the last 3 days, that yes, the coercion, it does account for a proportion, but not a particularly big proportion. It's not 50% of them. It's, you know, it's a small proportion. So people are coming out and looking for help at greater rates, irrespective of coercion.
Time for morning tea. Thank you very much.
Thank you.